day 2 of CTMB. I'll start by congratulating John Hopfield, who got the Nobel Prize this morning for physics, and we have one of his students right here in the front row, Mr. Onuchik. So it's very exciting that the father of the family got awarded the prize today. So with that, let's get started. Our first speaker is Garegin Papoian. As Vitor said, a lot of people here are in the family scientifically. So Gareg was a CTPP postdoctoral fellow over in San Diego, and then has been, we've all seen how successful he was with Peter's talk and other talks. His name keeps popping up on a lot of the slides, so I don't need to say a lot about what he's done. But after his postdoc, he was at University of North Carolina briefly, and then University of Maryland, where he's an endowed chaired professor. But that is not enough for him, so now he's going to go take on Google, I believe, and take down the AlphaFold, right? So he's shooting big, and I'm looking forward to hearing what you have to say.
Do I put
Oh, yes.
it works? Okay. Yeah, I have, this is the printer, yeah. Yeah, thank you, Paul, and thanks for the invitation. So simulating life processes across scale, it's a grandiose title, but I have worked across scales just over my career. My PhD was in quantum chemistry, and I worked all the way from molecular dynamics to coarse-grained modeling to cell scale. So there is some truth to this, and that's actually is what we are in this company, Deep Origin. That's what we are trying to achieve, and we'll see how it will work out as we go on. So my talk has two parts. One is about my role in the company or what we are doing there, and second is my work at the University of Maryland. The company works in drug discovery, so that's why you can sort of see this. In drug discovery, you have this sort of phases. You have to find and validate a target for a disease, and then characterize it, hit discovery, and then all the way to lead optimization. And we develop simulation software in all these areas, and that's why what makes it quite unique, because if you look at typical players, they tend to specialize just in one. We sort of are developing in at multiple scales, and in the future are hoping to connect them up in some multi-scale way. Today, I'll tell you, it says structural ensembles, but it should be coarse-grained modeling here from the perspective of this audience. So I'll tell you about this. So that's where Awesome comes in. And I'll tell you about virtual screening. We've developed our own molecular dynamics engine like GROMACS. It's faster than GROMACS. And we are working at cell scale too with experiments, but I won't tell you about that. So in the first part of my talk, I'll tell you about mesoscale modeling of the cytoskeleton and sort of our long-term program to simulating eukaryotic cells. So that has been 15 years of my work at the University of Maryland in College Park. And then in the second part, I'll tell you about our work at Deep Origin, and in particular, that there are some problems where AlphaFold greatly outperforms, Awesome greatly outperforms AlphaFold 2, and about our new algorithms in virtual screening and docking and so on, and machine learning. All right. So for the software and algorithm that we developed is called Median. So in the first part of my talk, I'll describe that, and you see here people and collaborators over a long time that helped to develop Median. There are lots of beautiful movies nowadays, super high resolution of how cells interact. In this case, you see actually an immune cell attacking a cancer cell, and I think there are we'd like, and there are other people, including in this audience, that would love to see these kind of processes being simulated from physical chemical principles. And it's still sort of, we are not there yet, but getting there. I think that will be very exciting, and it will change actually medicine, because then, because until now, biology, that the way people think, or people think about diseases and so on, it's very qualitative. If we had quantitative models, then that could dramatically accelerate drug discovery. And I'd like to give you a sort of small background on that, so that sort of shows that there is hope. This is a landmark paper from 12 years ago, in 2012, from Marcus Covert's group at Stanford, and Jonathan Carr, who was the first author of this paper, he works at Deep Origin, and they used a tiny bacterium, or they worked on a tiny bacterium, mapped all the genes, I think several hundred, and wrote an ODE model that they parameterized, and then they made it quantitative. So it was, they could show that by making various perturbations and mutations, they could predict what the phenotypic outcomes are, and then did experiments and showed that. So it sort of showed that it is possible to predict cellular dynamics. It was a zero-dimensional model, no mechanics, no geometry, no structure, but it shows that it's potentially possible. Eukaryotic cells are immeasurably bigger than that and more complicated. So so that work just shows that there is hope, basically. My work has been for a long time at the cell scale, motivated by the cytoskeleton. I'd like to show the slide to show that cytoskeleton is complicated, and this is a highly simplified version of what goes on in the cytoskeleton, and also it's a dated slide, so it's about 20 years old now. But you have proteins, so you have polymerization, depolymerization, branching, severing, acceleration, deceleration, like all sorts of regulation that you can imagine. And it's an intricate system that self-assembles in one part of the cell, disassembles, it responds to external cues, and it's involved. It's sort of, I sort of think of it as one of the brains of the cell. In addition to, like often when people think of cytoskeleton, they think of it as mechanics, like forces pushing cell shape, but it also integrates signaling and so on. And so that's why I think of it that also it's a significant part of information processing by the cell. And we've been interested in modeling this at a higher structural resolution than being done before, because previously most work was at the level of a two-dimensional PDE model. People who do it would model it in MATLAB, like a simple models like that. Still very insightful, but we were interested in structural physics-based model. On the other hand, the scale is humongous. We cannot really have amino acids and so on, so it has to be coarse grain. So coarse graining is roughly at the scale of 1 actin monomer is 1 bead. And we have a two-phase model where we have this cytosolic background where we are going to do stochastic reaction diffusion, and also have polymeric part where we have we have polymers connected and forces. I'll show you more on that. Molecular motors that both can walk on filaments or can exert forces on filaments, things like membranes, all the things that actually you think at the cell scale are important. And here you can see the cytoskeletal reactions that we include. As I said, capping, polymerization, depolymerization, branching, and so on. For the cytosolic phase, we do we simulate reaction diffusion master equation using various Monte Carlo techniques, and again, the idea is to be able to resolve spatially how molecules are moving around and reacting with each other. And we can put arbitrarily complex reaction networks, so we could have hundreds of various chemical species interacting in complex ways, although we typically would use something between 5 and 10 chemical species. This slide shows a little bit of detail of both the cytoskeleton and the reaction. So for example, you have, if there's an polymer end and an actin monomer, it could associate or dissociate with certain rates, and the stochastic dynamics has to model that correctly. Or there are molecular motors, they can bind and unbind. When they bind, the heads can start walking and generate sliding forces, and there are cross-linkers and so on. So it shows that there's both chemical kinetics, but also you can see structure and also mechanics, like all these things very closely interact. The mechanical part, so in addition to chemistry, this reaction diffusion master equation, we also need to have mechanics. And it turns out that actin filaments are stiff polymers. The aspect ratio, if you look at the persistence length divided by the radius, the persistence length is about 20 microns. Radius is between 5 and 10 nanometers. So the aspect ratio is over 1,000. And We can model it with beads, but it's a very wasteful representation. So we actually end up modeling it with cylinders connected at the hinges. I'll tell you another representation in the next slide that's more fancy. But let's sort of, most of the time we model this because it's computationally more efficient. And then we have to have other potentials. For example, you put a bending potential so you can correctly describe the bending rigidity of actin filament, exclude volume, and so on, how motors bind. The fancy representation is this Kossler-Rode model that instead of having this cylinders connected at hinges, it's a continuous, actually, imagine a continuous line and then it's a tube. So you describe it using splines, and then when you do that, then you can also model shearing, twisting, stretching, and bending of filament. It's largely an overkill for most problems, but in some cases it might be important. In particular, twisting of filaments can be important. And so for those kind of problems where you do need this sort of extra degrees of freedom of filaments, then we can also have a more detailed description of actin filaments. Also, we developed a membrane model. Membrane model is a parametric model where we simply parameterize a surface imagined by dots connected up like this, and then based on discrete differential geometry, we can compute curvature, area, local areas, and so on. And from that, we can then compute the tension, bending. We can also include osmotic pressure because we can compute volume and so on. And then in addition, we're going to put the cytoskeleton there, so then you have to have also this localized interaction between cytoskeleton and the membrane. In addition to that, so these interactions, we also discretize or think of the membrane as having these compartments for reaction diffusion. So we can have reaction diffusion then separately on the 2D manifold and 3D manifold. The reason for having separately on the 2D manifold is to simulate dynamics of things like receptor proteins. So receptor proteins, membrane proteins, curvature-inducing proteins, they can diffuse around, they can cluster, and so on, interact, change membrane shape, interact with the cytoskeleton. They can actually hop on and off, so they can desorb to the bulk or absorb to the surface. So it sort of gives you both separately stochastic reaction dynamics, both on the membrane and in the cell cytoskeleton, or in the cell cytosol. And then the final part is we use a little bit unusual ansatz for kinematics or dynamics. I already told you that we simulate with Monte Carlo, we simulate the master equation, so that's what we do with chemical reactions and diffusion. For the structural part, a more natural thing to do is to do Langevin dynamics. But because these are stiff networks, if you were to do Langevin dynamics, most of the time you just see vibrations, because think of it like, again, it's almost like this metallic rods connected up. And it turns out that there is this timescale separation between chemistry and mechanics, and because of that, it's advantageous to do the chemical dynamics, and then the mechanical equilibration is slave to it, and we can sort of make arguments for why that's valid, and that allows to accelerate simulations to by about 100x, and the reason we want to accelerate simulations is we'd like to simulate human volume cells at about 1 hour timescale or half an hour timescale, something like that, something that you cannot do in a coarse grain model. And this slide shows both the sizes of, it sort of shows that what happens if you sort of go to like these different volumes, like here the last line is about 100 cubic microns. Human cells are about 1,000 cubic microns, and you can see that we are getting there. That there's a new version of Median completely from ground up written in this new programming language, Julia, which for those of you that are interested, talk to me after my talk. But it allowed us to greatly accelerate it, and it also made it much easier to develop and so on. And this shows one example where we can you can see we're interested in axonal modeling. We have a collaborator at Ginniger at NIH doing imaging and working there. We've had a number of papers with him on this. And you can see that if you sort of go to this size, like it's about 7 micron in length, there are about 300,000 actin monomers in this volume. And and it takes us 3 days on a single core to simulate this. It's not the program is still not parallelized, and you can see the simulation time, which is 1,600 seconds. So it sort of gives you the scale, and you need to go to this both large scales and large both length scales and time scales where interesting things happen biologically. The software is the C++ version of the software is available. We are planning to write up. We're in the process of finishing writing up the Julia version and also making it available. So I'll show you some results. Quickly, we've done a number of both internal work and collaborations to use Median over the years. This work was focused on sort of bringing this membrane model and thinking about, for example, membrane protrusions. And you can ask interesting questions. So if a bundle makes a protrusion, what are the conditions for making a successful protrusion? For example, how thick should be the bundle? How many filaments? Because if it's too thin, it will buckle, right? And then how about the angle of attack? For example, what if it comes straight to the surface versus sort of a certain glazing angle? And then depending on that, you can see that you either make a successful protrusion or not. And so a number of questions like that were answered in that paper. And here you can see video sort of showing the complexity inside. Here's another version that if you put another geometry inside, then instead of making a protrusion, you have something going on there. It's again micron scale. And this actually, at least in principle, tells you that if you were just to make this more complicated, include hundreds of genes and receptors and so on, that this could be the way to sort of to get to modeling eukaryotic cells. So couple of scientific stories. So one work was on this idea of entropy production and avalanches and actomyosin networks. So Kel Floyd worked on that. It was a collaboration with Chris Jarzynski in my department. And so we were interested in actually writing up. It turns out that quantifying entropy production was really non-trivial because this is a small system, and some of the thermodynamic formulas needed to be, let's call it, adapted or worked on to get it right. Otherwise, even the sign was coming out wrong if you just use textbook thermodynamics. So we managed to get it right, and then we sort of saw that in addition to sort of what would be like normal Gaussian fluctuations of and so on, there were these log normal. There were these like there was this fat tail, and in this paper we quantified that, and we sort of proposed that there are these cytoskeletal avalanches. Margaret Chang and Peter and Herbie and several other people, they had a really nice paper on avalanches more from structural perspective in the dense dendritic networks, sort of again showing that there are these sudden movements that you'd call avalanches. And there are experiments showing that actually experimentalists about now 8 years ago saw something that they call cytoquakes. So if you put a cell, there are these beautiful experiments from Johns Hopkins, from Reichel group, where if you take cells, put them on micropulse and monitor micropulse motion, then again, you sort of see these Gaussian motions, but sometimes there are these spikes, and they can be quantified, and again, that they see these heavy tails, they sort of see something similar to the Richter law, so that's why they are called cytoquakes. But it wasn't clear from these experiments I mean, most likely it's cytoskeleton, but other than that, it wasn't clear what was causing this, what's the origin of it. So we, again, we sort of worked on this question afterwards, and in particular in this work, we did Hessian analysis of the network, and we were interested both in we're interested in the question like, could you actually predict a cytoquake? If I look at the cytoskeletal structure and do some analysis from actually machine learning to physics analysis, can I know that in a few steps it will undergo a cytoquake? And the answer is yes, you can, although structurally it's identical basically to non-cytoquake states, so you have to do something like this Hessian analysis and some other things to get there. And we saw that there's two types of modes, sort of stiff modes and soft modes in the network. and that the sided quakes were mediated by the soft delocalized modes. Interestingly enough, so these were actually non-dendritic systems. In a recent work, interestingly enough, we are seeing that in dendritic systems, maybe it's the other way around. So it's an interesting question, just there's lots of detail here. And this new work is a collaboration with Michael Morrell at Yale. They are doing in vitro assays of actomyosin systems. Inspired by these works, they decided to look at these avalanches and cytoquake-like behaviors, and they do see that there are these non-Gaussian tails in the systems, and we did computer simulations, and these are dendritic systems, and where you can, by changing RT3 concentration, you can vary the dendricity of the network and so on. And we get semi-quantitative agreement with the experiments, and again, we sort of see that this is a fairly general phenomenon in that sense. This is a manuscript that's under review right now. All right, in terms of some of the newer stuff, we developed a more detailed myosin model. So for those of you that use Median, like myosins, actins used to have excluded volume, they wouldn't cross, but myosins, for whatever reason, we had sort of a very coarse-grained model of myosins, but now sort of we have a myosin model with multiple heads working, and in particular, if there is an actin, for example, here, then as you saw, that myosin would pause, and there are experiments actually showing that. So if you see that there are experiments showing that the myosins, when they sort of see a junction, they sort of get stuck there for some time before they go. So we are trying to, at least in some parts of the model, try to make it increase the realism, if you'd like, and increase more detail. And the last part of applications, we have also been interested, although not only now this work is coming to fruition, we've been interested in modeling dendritic spines. And for one of the questions I was interested in, how do you get this mushroom shape of dendritic spines? Can we have a model that would sort of produce it spontaneously? And dendritic spines, so because of Peter's talk, I'm not going to give too much introduction to it, just to say that there are all these various cytoskeletal components, and there's also spectrin that's really important for this shape thing. There is coherence. So we put all that in the model. And you can see that when we do put, and you can maybe see it here, that at the top we have to have coherence that's sort of connected to the stuff outside. We have branching effectin, you have linear effectin, and then you have spectrin. When all these things are put together, then you can see that the simulations then produce this. It took us 5 years or 3-4 years to get there. So it's very non-trivial to get this mushroom shapes, but we were able to have enough ingredients to produce spontaneously these shapes, and in particular you can see that inside you can see sort of the dendritic cytoskeleton at the top, but it turns out that the spectrum here at the neck is also important, and that was already clear from Tatiana Svitkina's work using beautiful electron microscopy measurements. So it seems that we made progress with this, and we are writing this up. All right. I'll just shout out to also this paper where we're in general interested in the question of emergence of these rings and cortices. Most eukaryotic cells have an actin cortex that's about 100 nanometers to 200 nanometers thick. Why they are there it wasn't very clear, so we had a paper where it was a combination of experiment and simulation to explain that question. All right, now switching now to the deep origin part of my talk, and you can, Michael Antonov, he was a former co-founder of Oculus, the virtual reality glasses, and so he has, interesting enough, he's a computer scientist from UMD actually, a UMD alumnus, but he has passion for simulation, so that's how we sort of got sort of partnered up. And here I'll just say, many of you know Aram Davtyan, who was a graduate student with me. He was the lead developer of the awesome software back in the 2012, and then he was a postdoc with Peter, and then he worked at Atomwise. So really great career, and he joined us a couple of years ago. All right, so I'll talk about Awesome here. Again, I'm not going to give too much introduction. In this part of my talk, I'd say that I'd like to make sort of comparisons to these ML models, AI models, that it's not just AlphaFold, there's a whole zoo of AI models, and the question is like, have they solved all problems? Like, you don't need to, maybe you don't need to know physics anymore, you just like throw data, these models just digest it, and the answer is no, we are not there yet, and again, we can go through the reasons why. And there are many, like if you talk to real biologists or in drug discovery and so on, there are many complex things that really cannot be modeled by these AI models. AlphaFold 3 claims that they can at least to model some of these, but I think it still has to be seen what exactly they can model. Their paper was more of an infomercial than a real paper. I have this slide, again, I'm happy to share this later, so I don't want to spend too much time on it. But there, as I said, there are AlphaFold Rosetta models, but there's an ESM fold, and they are not the thing. Like these are large language models, and these are like this transformer model. So they have pros and cons, and this is actually a slide that are unprepared. Again, that there are in certain areas where they do amazingly. These models do amazingly well, and they are widely used. But also there are limitations. All right, coming to Awesome. So at Deep Origin we did some work on Awesome. So it's let's say there are certain improvements, nothing too dramatic, but there are some significant improvements. And for structure prediction, you can see that for fairly complex proteins, we can get to I think for proteins up to at least 200 residues, we typically would get something like this when we use ER, evolutionary restraints, which is the same thing as AlphaFold uses, which is MSA, right? I'd like to focus on binding. There was a paper from Peter's group, and we collaborated in 2012. We took the same proteins from that paper. So these are all dimers, and you can see sort of predicted versus crystal structure, and we compared sort of this reworked version of Awesome, and you can see that we don't get this is Q. So we have some improvements. I'd say moderate improvements. But we worked 2 years on different aspects of Awesome, and I'm happy to have an offline conversation about it. One result I'm excited to share is this. So there is this new class of drugs called PROTACs, where you might or might not know that 90% of human genome is undruggable, or at least has never been drugged. Not human genome. 90% of disease-causing proteins have never been drugged. So we only so far drug 10% of things that need to be drugged. And many cancers and so on have proteins that we know that need to be drugged, but we don't know how to drug. These PROTACs give hope that some of them can be drugged because basically they are dumbbell-shaped molecules, and they connect they connect your protein of interest to an E3 ligase, which sends it to the degradation machinery, and these anyway. So this is a new class of very hot area in drug discovery, and you can sort of see it here that there is some sort of, again, you have a protein complex forming, and there will be a drug there, and you can actually see that drug here. And what Aram did, he took 12 such complexes from the PDB that has recently been worked on, and he removed the PROTAC, which is that drug, just 2 proteins, and he predicted the structure, and he sort of looked what's the best structure you get out of this compared to the crystal structure, and in one of the best cases that we have, you can see that we can get like a really complex sort of system. We get 1.5 angstrom RMSD. You can again see Q. This is Wellness Q. If you have there's a question about that. And this is the slide. So we can do the same thing with AlphaFold 2. We haven't tried AlphaFold 3. And this is RMSD. And ideally you want to be 1.5, but maybe from 2 to 3, like in this range. You can see that with Awesome, except for these two, like all the rest are sort of in that range. And even these two are like 5 to 6. But if you compare, you can see that only in one case there's a similar performance, but in other cases, when you are at that 10 angstrom RMSD, yeah, there isn't much usefulness coming from that modeling. So I think this sort of shows that the reason that here we greatly outperform alpha T is that these are non-native interactions. These are complexes that were not meant by nature to interact because they don't in the cell. These are artificial constructs, right? And AlphaFold 3 is, I think, over memorizing things. And here in this case, where there's nothing to remember from or recall from, it fails. But Awesome is largely a physics-based model, although it has Hopfield nets in it somewhere. But I'd say that most of Awesome is largely physics-based, and that's why even for these non-native interactions, it does a good job. All right, virtual screening, which is another problem, docking. I'll just show you some results. We developed our own docking algorithms. It's a combination of physics and AI. It turns out that you need both to get to really good performance. If you ask a question, okay, if I'm taking a small molecule, try to dock, what's the, and I look at the top state, what's the probability that I'll get less than 2 angstrom RMSD structure? You can see that with Autodoc Vina is one of the most widely used docking software in the world. It's open source, and we can see that you get 50%. We are now around 72%. So it's really hard to get it to those scales. And for the docking problem to be solved, it has to be probably 95%. So it's still lots of work to do. AlphaFold 3 results. If you look at all the PR that Google did, they all said it's 75%. If you look at the paper, all the LinkedIn posts and everything, New York Times, that was the thing. You go to the SI of their paper, and in the SI they have this thing, and they say that, oh, if the protein is not similar to our sort of training set, and ligand is not similar, then we get 49%, similar to Autodoc Vina. So these are the games that people play, and you have to read SI, apparently, to see that. So we are doing much better than that. So. In addition, so in docking there are two problems. One is structure prediction, another is energy. Autodoc Vina, you can sort of see the prediction of energy. It gives you about 62% correlation coefficient. With using the machine learning that we developed, we can get to about 90%. So we also developed AI models that can predict other chemical properties of molecules like solubility, lipophilicity, various types of toxicities. There are like 15 types of toxicities and so on. And then we compare them with other state-of-the-art models, and here it's sort of you want to have small error. I'll skip that and just show you. We also developed an AI assistant, so I'll leave you with that. And the idea of AI assistant is that, especially for medicinal chemists who are not prone to using or sort of have an aversion to using computational chemistry software because they are complicated, you can just talk to the AI assistant and it can do, it can fetch proteins, ligands, do docking for you, things like that, and hopefully can it work? Oh, you know this, sorry, I don't know, that last movie doesn't work, but you can even sort of say, so what do I do next? Let's say if you want to work with COVID, it says, oh, download this protein, find the pocket, it can guide you actually and tell you what to do, but under the hood it runs the state-of-the-art drug discovery algorithms, either ours or we also have another 20, 30 software there that are open source. So it's even, I think even educationally it's a great tool, and in a few weeks we are going to release it publicly. Okay, thank you very much.
Excellent talk. And I'll just say I really like your point about telling people to read the SI. I hope all of the students take that point really to heart, because you will, at one point in your career, say, I know this because I read it, and then your advisor or some colleague will say, well, you don't know what you're talking about. And then you go read and you'll see, oh, yeah, I didn't actually pay attention. So if you're young, don't make that mistake. It's a great lesson. So, any questions?
It was a wonderful talk. I'm curious about the dimer question, something that is dear to my heart. I think dimers are very important for many, many reasons. However, when you put evolutionary constraints, something that's also dear to my heart with DCA and all that, there's very little data, evolutionary data, when you go to dimers. We notice that you do very well for proteins, but base evolution data. Have you noticed that? What do you do about it? Is that something that's going to really limit?
So that's what's great about all this awesome work is that we don't have that actually. So we have ER evolutionary data for the monomers. We can even use AlphaFold to put that ER like constraints on monomers. But for the dimers, we have nothing. Just it's pure awesome potentials, the water-mediated potentials basically that Peter and I worked on back 20 years ago. And that's enough to give you those results that you saw.
And is that enough, or is there? That's what I'm curious. You get all the possibilities just doing that,
So I'm sure. I mean,
because AlphaFold try to use evolutionary information for dimers, and that doesn't work that great.
So in this work, we didn't use it at all. Like there was zero evolutionary information for dimers. If in some cases you do have it, if we put additional ER there between dimers, I'm sure it will improve the results. As you saw, for most proteins, we were getting between like about 2.5 to 3 angstrom RMSD structures. So there is, I think, so there is room for improvement. Like you'd like to maybe push it down to 1.5 or 2.
But I don't think evolutionary data is going to help that much. But anyway, okay.
Yeah.
Hi, Garrick. Hi.
Yeah.
Very nice. I had a question when you were doing the master equation solution with Gillespie. Is that still well stirred, or are you thinking about the spatial arrangement as well?
That's a good question. So I went a little too fast. We partition space into compartments, and compartment size is typically several hundred nanometers. So within a compartment, we assume that it's well stirred, but between compartments, we have diffusion. So you can have spatial heterogeneity across sort of, typically we look at, let's say, 5 to 10 micron length scale, then you sort of can have this spatial heterogeneity with that sort of level of voxelization.
I have a question about the full cell simulation or the filament simulation. Like these are a lot of players, and everyone have their own kinetic parameters, and the space is very big.
Mm-hmm.
How do you deal with that? Like, do you use only experimental data, or do you explore it?
We largely use experimental data because actin has been a system that has been studied for decades. Probably many billions of dollars of research went into it experimentally to measure all sorts of rates under all sorts of conditions. And so many things have been measured. There are some things that haven't been, and there we sort of try to guess. You can still have reasonable guesses for something similar. But yeah, so we largely in these actin systems use experimental kinetic rates. I think so. I mean, this kinetic rates data is more like an input to the model, but then when we, for example, in this actin ring paper or this avalanches paper with Michael Morrell, when we make certain predictions and then compare with experiments, when we see that things at least are semi-quantitatively in agreement, then we sort of think that, okay, maybe the model and all these rates are reasonable. But definitely there is room, I mean, there is quite an uncertainty there in the sense that there are certain things, like not everything is pinned to a well-defined value. Yeah, so that definitely is a good question, and we struggle with it.
Are the parameters in vivo the same as in vitro? Because I guess many parameters have been measured in vitro.
Yeah, many, and there are differences between in vitro and in vivo parameters. So again, we sort of know that, so for example, this actin ring paper, that was the whole point of it, is that actin turnover dynamics is faster in vivo than in vitro. So if you put in vitro rates, you don't get these rings, but if you put in vivo rates, you do. And we sort of explain why, but also just that observation that actually it's about a 10-fold difference there, and that changes physics completely. Yeah, so definitely, yeah, as I said, important question.
Any more questions? Well, if not, thank you again.
Thank you. So this is going.
Garrick, do you have a pointer? Ah, thanks. So, we have 45 minutes till we go to the 30. It's not a problem. Okay.
Yeah.
So, our next speaker is Serdal Kirmizialtin from NYU Abu Dhabi. He was a grad student at UT looking at developing advanced simulation techniques and hand sampling methods before he went into high performance computing as a postdoc at Los Alamos, where I also was, but not at the same time. So we got to meet later, which was a lot of fun. And since then, he recently got tenure in the chemistry department over at NYU Abu Dhabi, and working on a lot of single molecule properties, you could say.
Right. Thank you so much, Paul. Do you hear me well? All right. Thank you, and thank you for the invitation. It's a great pleasure to be in Brazil. I haven't been here before, so I'm very excited. I have one-day excursion activities afterwards, so I'm looking forward to see the city. So my research is basically on studying all-atom simulations, the RNA structural dynamics, and RNA folding problem. And the way that we currently study this problem is in collaboration with experimental people. We try to, there are some limitations I'm going to explain in a couple slides. We try to couple experiments and simulation to develop a consistent framework that allows us to basically physically to represent the atomic scale, how the nucleic acids fold and function using all-atom simulations. Okay, so the justification is very obvious. RNA is one of the essential elements of life, and it touches in every aspect of the cellular processes, including translation, transcription, gene regulation. RNA is also very important for therapeutics, and we have been looking for druggable RNA molecules to expand the pharmaceutical solutions to the diseases. So, okay. But RNA is also at the same time a very interesting physical problem, and it has three types of major governing forces. One of them is the base pairing through the hydrogen bonding. That is sequence specific, and it's a short range interaction. And the other is a hydrophobic effect, which is basically through the stacking of the base pairs nearby to each other. And the third one is the electrostatic forces that is basically governed by the counter ions in the solution and the highly charged nuclear base phosphate backbone that interacts with the counter ions. The interplay between these three major strong forces, one of them is long range, the other two are short range, leads to a diverse set of conformations and the complex structural geometries that we see in the protein data bank. The current studies of structural properties of the nucleic acids heavily rely on a base-centric approach, which relies on how the two base pairs fold each other, try to predict the folding, and try to understand the folding mechanisms of RNA. So we argue that this picture is not sufficient. A biophysical balance view is needed to be able to understand the RNA. And this basically allows us to understand how a four-letter code that creates so diverse structural complexity, and we will be able to understand the main players governing the folding and changes in the folding in the presence of different solvent conditions, and also basically to be able to control them and to be able to visualize their atomic structures in solution. So the challenges in RNA, studying RNA, I divided into two groups. One I call the experimental challenges. So typical methods used for proteins, which is basically structural biology, crystallography, cryo-EM, hardly works for RNA systems because of the dynamic nature. And RNA molecules are very sensitive to the solvent. That sensitivity creates a huge problem for the cryo-EM or crystallography. And RNA molecules fold and function in the presence of the cations, and this unique interplay between cations and the RNA is an important component, which is almost impossible to measure the influence and the details of these interactions through the experimental approaches at the moment. There are solution studies, unlike the cryogenic methods available, but these solution studies, like the FRET, small angular excess scattering, are limited in resolution, and the signal comes from an average structure which does not provide details needed to understand the RNA systems. Computer simulations, on the other hand, I'm only concerned about atomistic simulations. The problem in atomistic simulations is that the conformational space is a lot wider, and a monomer has about 7 degrees of freedom, as opposed to proteins, which has 2 major angles of that we need to concern. And the energy landscape is a lot rugged because of the strong base pairing and hydrophobic interactions. And current force fields suffer from imbalances that basically makes the all atom and beam simulations provide an inconsistent picture on the RNA structure and dynamics. So for about 5 years ago, we decided to basically start to couple experiments and simulations to limit the limitations of these 2 methods, and then to join these 2 forces together to create to develop an understanding of the RNA systems. And the idea here is basically that experimental systems create some provide evidence to cure or fix the problems of the empirical potential and sampling, and the atomistic simulations provide atomistically detailed description of the system. So these 2 forces, when they join together, they can provide us some understanding of the nucleic acids. So the way that we do is we use maximum entry approach for coupling the simulations and experiment. Ideally, let's say that we have a measurement that we would like to observe in the simulation, and this measurement can be obtained through the classical solution of the Newton's equations of motion, and then computing this through the averaging over the ensemble. And the value that we get, we call it F0, that comes from the unbiased probability density function of the empirical potential, may not be correct and may not be giving us the right answer that we are looking for. And so if that happens, we would like to basically seek for a probability density function that is biased towards the experimental signal. That's that when I create this probability density function, then canonical averaging of this ensemble gives me the experimental signal. So this can easily be done by maximizing the Gibbs entropy of the bias potential, instead of using the 2 constraints that we have in a canonical ensemble, we add another constraint which basically satisfies the experimental signal. And we hope that by adding this constraint and then solving the following equation to find the highest entropy, we ended up a distribution. And analytically, this distribution is nothing but the canonical distribution plus an addition of a linear term that basically computes the observable in the phase space. And this linear term becomes a bias added to the molecular dynamics simulation, and that allows us to basically go from the unbiased distribution to a biased distribution that meets the experimental data. So SACS is one of the most comprehensive experimental techniques that can couple with molecular dynamic simulation. And that's the reason why we decided to use this as a method to integrate the molecular dynamic simulation. So in this, the advantages of SACS is basically it is measured in physiological conditions. Basically, it reports on an RNA that's in its native environment. And it can access to the ensembles rather than providing us a unique structure. So it's SACS and WACS, when combined together, it provides us a long length scales and the short length scale correlations. So we can look at the long range and short range details of the atomic structure. And also, in addition to the RNA conformations, we can provide some evidence about the ionic environment and the solvents around the RNA molecule. So it combines all these different informations into one single signal, which makes it a good way to basically couple with an experiment, with a simulation. So here is the machinery. Basically, like we run MD simulation, solves the equations of motion, and then from the positions, we compute the scattering amplitude. Scattering amplitude for the solid system, which includes the water and ions, and also RNA. And then we have another simulation system, which we basically compute the scattering amplitude coming from pure solvent. And then the difference between the two, average over ensemble and also orientation, gives us basically the scattering amplitude curve that comes from the simulations. And then the biasing term, the linear biasing term, basically moves this ensemble to the probability density that meets the experimental signal. So the way it works is basically we start with an initial guess of the structure based on some bioinformatics tools, and then we run an MD simulation, and then we watch our conformations go from the initial state to the final state, and then we collect the data during this step, and then we observe how the conformations go from basically a state where they are far away from the experiment to the state where they meet with experimental data. And the accumulated data then used to create a machine learning model. That machine learning model basically later we use to find what structural features are hidden and coded in the signal about from the wax features, we can basically interpret the structural features. So using this technique, we looked at various fundamental units of RNA. We looked at double-stranded helices, and we also look at single-stranded helices. We look at some biologically important RNA molecules. And I'm going to share a few results of these studies, and then to show like how this integrated approach is helping us to understand these molecules better. So the first example I'm going to show is basically what is the structure of a DNA helix in solution, and how does this structure changes with salt conditions and sequence, and what is the structure of an RNA double helix in solution and how does this change in changing salt conditions and sequence. So to answer this question, we first basically do our machine learning and find out that the signals that we see in the SAX profiles, what kind of information they provide us. This is an important thing to be able to interpret the results. And what we see is that as far as the double helices is concerned, the signals that we see, like highly correlated with the phosphate-phosphate major backbone distances and helical conformations like helicity or the helical information, helical radius and the twist. And these are informations like the base pairing geometries or relative orientation of the bases are not seen by small angle X-ray scattering. So this, based on this, we look at. Okay. So here is the two DNA systems. So when we look at the two sequences, one we call the mixed sequence, which has equal number of nucleobases. The other one is a polymeric sequence form of A and T. So here is basically the fit coming from simulation and experiment. The lines are simulation and the dots are experiment. And you can see that B form, like the mixed DNA and the poly-A, are structurally different from each other from the SAX profiles. And the difference are due to, according to our mapping, comes from the machine learning, are due to the differences in the major groove and minor groove width. So bDNA in solution is forming a B-form DNA that is confirming the crystal structures. But poly-AT, on the other hand, has a very narrow minor groove and a wider major groove that basically deviates it from the B-form structure. So basically, the sequence makes a huge differences in how the major and minor groove are partitioned on the helices in the case of DNA. When we look at the salt effect, on the other hand, like this mixed DNA and in MGCL versus in KCL, we don't see any difference between them. So DNA is sort of insensitive to the solvent conditions, but it is sensitive to the sequence information. So when we compare a DNA with an RNA, we see a dramatic change in the signal profile. The SAX profiles are dramatically changing. This can be understood because we know that double helices of RNA are A form and double helices of DNA are B form. But what was surprising to us is that when we change the salt conditions in an RNA double helix, we see a dramatic changes in the profiles. And a same sequence is in KCL versus in NaCl versus in MgCl shows a signal that is kind of different from each other. And these differences can be resolved through the correlation maps. And this basically gives us the following picture. So a DNA, an RNA, in the presence of MgCl has a major groove width changes dramatically from the A form structure. So none of the RNAs that we study show basically an A form structure as it was proposed by the crystal structures that we have. RNA in solution deviates from the A form, and duplexes that we study show variation depending on their salt conditions. And in the presence of KCl, we observe a bending of the DNA that is resolved by our integrated approach. And this bending is slightly less in the presence of NaCl, but it's not present in the case of MGCL, which basically says that the structure of an RNA are coupled with its solvent environment. So even in the level of a duplex, which is supposed to be like one of the most rigid elements of an RNA, there is a huge sensitivity of the salt conditions, which is not present in the case of the DNA. So why does the RNA bend in the case of monovalence? Now we can ask these kind of questions because we have an MD simulation. So we can go back and then analyze our trajectories. And from the analysis of the trajectories, we construct ion densities around counter ion densities and then observe the differences. Here I'm showing a DNA, and then the DNA and the counter ion cloud that is forming around the DNA. So you see that for AT-DNA, we observe 2 layers of binding. So one of them goes above the phosphate backbone of the minor groove. The other one goes through the major groove. So they're very well defined in the case of poly-AT. They are discrete in the level of the mixed sequence. So they have some sequence specificity in the case of mixed DNA. But we have 2 layers, which is kind of a unique property that we found through this study. An RNA, on the other hand, shows quite different ion binding pattern. So the minor groove is almost unoccupied, and all the density, all the counter ions accumulate on the major groove. So 1 layer condensation on the case of RNA and 2-layer condensation in the case of DNA is basically a major difference between the two, which might suggest like why the DNA is prone to solvent effects, but RNA is very sensitive to the changes in the solvent condition, which I will explain in the coming slides. So the reason why we asked the question like why an RNA and DNA shows such a 2 different pattern of counter ion condensation, despite that they have pretty much the same kind of charge on their surfaces. So what we realized that if you look at the structure of a double-strand DNA, the most negatively charged phosphate oxygens are facing towards the 2 opposite ends of the grooves. So one is basically facing to the minor groove, the other one is facing to the major groove. In the case of RNA, on the other hand, both of them are facing to the major groove. So these 2 oxygens of the phosphates that's facing to the major groove, creating a binding site that is unique to the RNA. And as a result of this, when we put the RNA in solution, and depending on the sequence, so for example, here we have a sequence that has high affinity to the potassium ions, and the places where there is a affinity to the ions are creating plasticity on the structure, bending the structure in different places, depending on the sequence information. So RNA, in a sense, is reading the counter ions and then changing and modulating its structure accordingly. So this finding is also interesting, which that this major groove binding is not non-specific. It also depends on cation size, which is we call the charge density. It also depends on the valence. So for a magnesium, we observe that there are two helices intertwined to each other in the major groove. So helix, the RNA helix, and then major groove, we have two other helices that are intertwined to each other. And these intertwined helices are merging in the case of potassium chloride, because potassium is a weaker binder, so potassium can shuffle between the major groove and the phosphate backbone, and then creating a continuous major groove. In the case of sodium, on the other hand, the ions are accumulated only on the oxygens of the phosphates. So oxygens of the phosphates on this backbone and oxygens of the phosphate on the other backbone, and then they jump between the two different electronegative sites during the simulation. So creating a completely two different mechanism of stabilization between sodium and potassium. We thought maybe this different means of stabilization has something to do with the RNA stability between sodium and potassium, because it is known that in the presence of sodium, RNA is more stable, RNA hairpins are more stable than in the presence of potassium chloride. So this study that was published like a while ago by the Bustamante and Tinoco's group using optical tweezers basically suggests that RNA screening or stability highly impacts by the charge density. So we asked the question, can the binding modes we see between sodium and potassium can be the reason why RNA is more stable in the optical spheres in the presence of NaCl. So to answer this question, we mimic the optical tweezer experiment. We mechanically unfold our RNA, and then we basically we looked at the free energy, the standard protocol. We look at are there any differences in the pathway of unfolding, and we saw that the transition states, the intermediate states, similar to the experiment, they are identical to both ion types. But what is different is the free energy of unfolding is about 9 kilocalorie less in the case of potassium chloride, which basically confirms the experimental result, which basically says that RNA in the presence of potassium is less stable. And this stability is also is close to the experimental result. The difference in the stability confirmed the experiment. So the reason for the stability surprisingly was not the fact that sodium ions are smaller, they can come closer and stabilize the RNA better. So that was the first picture that we had in our mind. The RDF analysis basically showed that the peak position, which shows the strength of the binding interaction, are identical between sodium and potassium. That is kind of surprising, but the reason for that is that despite that sodium is a smaller ion that can come closer, but sodium also has a strong solvation shell. So there's a competition between there's a competition of the water exchange between the ion and the oxygens of the phosphate that basically brings the interactions between, or the affinities of the two ions to be identical to each other. So sodium is not a strong binder to the RNA. That's not the reason for the RNA stability. So we looked at, and then as a result, when you look at the folding mechanism starting from an unfolded state all the way to the folded state, you see that the ions that are directly binding to the RNA increase as the charge density increases on the RNA, but at the same time they don't show any difference. The only thing that is different was the ions that are diffusively binding, that are not direct contact with the RNA. So what we surprisingly saw is that sodium accumulates more diffusive ions around it as opposed to the potassium. And so we asked the question why sodium is accumulating more diffusive why more ions are accumulating around the RNA when they are sodium, when they are not coming close to each in a direct binding. So what we found is basically like confirming the earlier expectation, is that sodium binds preferably to the phosphate backbone, while it rarely binds to the major groove. On the other hand, potassium binds weakly to the phosphate backbone, and most of the times it binds to the major groove. If you look at the surface areas of these two groups, you can see that the phosphate backbone has about 4 times higher surface area than the surface area of the major groove. So that creates a larger covering area to be filled, and that is accomplished by adding more sodium to the phosphate backbone surface rather than to the major groove. That basically explain why we observe more sodium ions around the RNA. That leads to the higher stabilization. So I discussed so far one aspect of the RNA structural dynamics, which is the cation distributions or the counter ions and their interactions with them. Another aspect which oftentimes ignored by this hydrogen bond-centric understanding of the RNA is the base stacking. And here is an example which is relevant to study this problem. So single-strand RNAs are known to be important biological signals, and they're important for many things, and they just got a Nobel Prize by the microRNAs on this field. So the question is, if these sequences are random and there is no major interactions between the bases, how come that the binding partners recognize them? So is there anything in solution when we look at this? Because crystallization of the single strands is impossible. There is no structural evidence that we can use to understand like what are the differences between these sequences that the biological binding partners can recognize and then bind them. So solution excess scattering provided us some interesting data that we use to interpret the conformational ensemble of these systems. And here I'm going to show how we did that. So typical in a typical sampling procedure, we couple the small angle excess scattering data with the MD simulation. On top of that, we add the FRET distribution, FRET signal as an additional experimental signal to avoid overfitting. And in our simulation, while we are basically running the Newton's equations of motion, we try to satisfy the Sachs constraint and also the FRET constraint simultaneously. This allows us basically to create a distribution that is lower, has more constraints that to satisfy. But at the end of the day, simulations were successfully able to meet both constraints simultaneously. And to check if the conformational ensemble generated by these signals are actually giving us the right distribution, we also cross-validated by measuring or computing a signal that is not used for the training. So we calculate the hydrodynamic radius, and then we compute the hydrodynamic radius for these single-stranded systems. And then we compared how well we represent them. So using this cross-validation and coupling the FRET and Sachs in MD simulation, we look at the dynamics of the systems. I'm going to play all of them. And do you see differences? sorry. This doesn't go back. Can I go back? Oh, that is okay. You saw my talk now. So, when a quick look at these three sequences, can easily see that there are major differences between these sequences. RA is forming kind of like a helical structure, and this helical structure looks like an A-form structure. RC, on the other hand, is also forming helical structures, but these helical structures kind of like deviate from the A-form. RU, on the other hand, is look like a random structure, right? So this, by visual inspection, we need to use Vitter's program to characterize it better, but this is our preliminary results. We see a clear differences between them. So we projected in some different properties. We looked at the base stacking propensities, and we see, again, RA is forming base stack conformations more than RC, and then RU is almost in the middle of a random coil and then slightly forming helical contacts. We look at the nature of these base stacks, whether they are A form or B form. What we realize that for poly A, they're mostly A form structures. The A form poly A is forming an A form helix, and poly C is forming a mixture of A form and B form. So there is a transition of the RNA going from the A form helices to a B form helices and then coming back. And this is the first time that in solution studies we have observed the presence of a B form RNA. And end-to-end distances basically also show like bimodal distributions corresponding to an A form structure and a B form structure in the case of poly C. And in the case of poly U, we see a helical structure, and a random coil structure that are basically mixed with each other. So the single-stranded sequences have some propensities, and these propensities also dramatically change when we change the salt condition, which I'm not going to discuss today, unfortunately, because of the time. So what we observe is that every sequence has an affinity to a different ion. And based on this ion, they can modulate the helical content and the randomness in the single-strand regions. So can we also study some biologically relevant molecules? We try to study this ribosomal RNA frame shifting pseudonode structure. So the reason why we studied this was a criticism by the reviewer saying that the RNAs you study has no relevance in biology. And what we are establishing is the methodology, and we wanted to show that the methodology can be equally applicable to study a biologically relevant RNA. And here we looked at a structure which was published 3 times in top journals, Science, Nature, and each time the structure keeps changing. So the sequence remaining the same and the structure keeps changing. So the question, like this one is a cryo-EM bound to the ribosome. This is an isolated cryo-EM structure, and this is the crystal structure. And so we asked the question like which structure is correct, right? So which structure is present in solution in the physiological conditions, so that we can use it for small molecule target. And the answer to this question was none of them correct. So when we compute the small angle excess scattering by like restraining these conformations and then running an MD simulation to create a solvation shell and to precisely represent the system, we find that they have huge variations from the solution conditions. And so what is the structure of the RNA? So we basically use our methodology, and starting from the structures that proposed, we were able to find a very good fit to the experimental data. And what we found is that actually it doesn't have one structure. It has many structures. And there is a chemical equilibrium between these states, and that creates the signal. So none of the signals alone, except for the crystal structure, are present here. But the RNA has a dynamic conformational pool that exchanges structures that gives us the signal. So that also highlights an important aspect of RNA systems, that there is always a structural heterogeneity that needs to be taken care of in understanding and studying the RNA systems. So far, we employ a method that relies on experiments. We need an experimental method, an experimental signal, so that we can do our simulation. So we ask ourselves, like, can we basically create a more reliable MD simulation protocol, so that one day, hopefully, we won't ask for an experimental data to be able to move on. And for that, we realized that this system is quite nice. This is called the helix junction helix. So it has very nice properties in the sense that it has the long-range interaction and the short-range interaction. It has flexible and rigid parts. It shows salt sensitivity. It has electrostatic, hydrophobic, and hydrogen bonding interactions. So it's a hydrogen atom of an RNA molecule. And this can be used as a benchmark or a system that can be used to basically test the performance of the force fields or develop new force fields. So current approach is basically using teeny tiny pieces of an RNA, which doesn't show most of these properties, and we call them, they're not real RNAs, okay? So, so this system can go from folded and unfolded conformations, and the way that we developed our force field is we used extensive sampling. So we ensure that our helix junction helix can cover all the conformational space accessible, and we achieve this using metadynamics. So we used a distance as a collective variable and angle as a collective variable, and then we sample conformations and we ensure that the system converge. And then once the system converges, we use the conformations to compute the experimental signal. And we rely on small angle X-ray scattering because we had very good data about this system. And here's so we compare simulation with an experiment. So our aim is, at the end of the day, to meet these two things after refinement of our force field. So before that, we tested like which force field is good so that we can start refining it, or are there any good force fields available. So we compare the standard amber force field for RNA, which everyone is using. And there's amber is a Dishaw Group's RNA force field, which was published as the near protein accuracy RNA force field. And then a force field developed by Aximantev's group called CUFIX, which is a version of an amber. So you see that the energy landscapes are completely different in three different force fields if full convergence is achieved. And the structures, the lowest energy structures, are also different. None of them look like the other one. And compared with the experiments, and none of them basically gives us a good agreement. Except HP CUFIX is slightly closer to the right answer, but it still fails. So we did a sensitivity analysis. We find out like what the problems are. We fix them. I'm going to just skip the steps. And then we ended up a new force field, refined version of the COFIX. We call them HP COFIX. HP COFIX allows us a much better agreement with the experimental signal. We tested with different salt conditions of the helix junction helix. We also tested with single strands of solution studies. It gives us the right answer. We also, one of our referees asked Bedir, can your new force field replicate the tetramer NMR couplings? And then we compare ourselves with the scalar couplings of 6 systems that, again, doing enhanced sampling through the UFET and then computing the J couplings through the Karplus equations. The green is the HP COFIX, which is trained using small angle scattering, but it gives much better agreement compared to the force fields that is trained using the NMR constraints. So rock force field, for example, has been using NMR to train the force field, but we use SAX data. We are accomplishing a better error in terms of representing the systems. We further refined the force field after doing this, saying that can we further improve it so that we get the experimental accuracy in the NMR restraints. For that, we again use the maximum entropy approach. This time, as a cost function, we use the deviation between experiment and simulation, where C is the parameters that empirical potential parameters, van der Waals parameters epsilon and sigma, and then we use this, we minimize this cost function to further refine our force field against NMR data. So HP COFIX was the version refined due to the SAX, and then RHB COFIX is now the refined version through the machine learning, through the NMR data. So we get slightly better improvement in almost all cases except for C, which our agreement was extremely good. But we ended up with a force field basically based on experimental data, and that now gives good agreement with solution studies. And finally, we tested this by 3 folding systems. We take an G quadruplex, and then starting from the folded state, which we unfold it, and then we calculate the energy landscape. And then once we calculate the energy landscape, we identify the top 50, the lowest 50 points of the conformation, energy points, and then we extract these conformations, and then we basically align them against the NMR data. And for the 3 systems we study, our system, the native state, the folded state, represents the NMR constraint, NMR structure accurately. So this basically suggests that experimentally guided simulations can also help to improve our problems in our force field. So to conclude, what I have shown so far is that the integrated approaches are the way to go so far in understanding the RNA and its complex interplay with the solvent environment. That allows us to, solution studies are the means to use to develop a better understanding of and the physics of the RNA systems. There are other players involved in the folding of the RNA, which are, aside from the hydrogen bonding, these are the ions distributions, the diffusive and specific ions. I was not able to talk about the specifically binding ions, especially magnesium. But there are other major players out there that needs to be understood to develop a better understanding of the RNA folding problem. And this allows us to basically understand the structural ensembles that is generated by RNA, and then to be able to fold RNA and predict their structures in the future. So lastly, I showed that integrated approaches can also help us to refine the force fields, to create force fields that is consistent with experimental data that can also be used for independent studies in the future. And with that, I'd like to thank my collaborators, Louise Pollack, who provided us, and we have been collaborating with her for about 8 years now on the small angle X-ray scattering. My PhD student, Wei Wei, who has done most of the work that I present today. Now he graduated and he's a postdoc in my place. And also Anya, who has done the RNA pooling experiments that I shared with you. And thank you for your attention.
I think we have time for maybe one question before we go to coffee.
That was a very nice presentation, and I guess lots of work, because I guess nucleic acids have been notoriously difficult to be properly simulated. I think this has been a decade problem, and it's always nice to see the amount of work there. My question is very, very simple. I think I would like to understand, why did you choose SACS data for that? I mean, because in the end you compare with NMR data. So why not use the NMR-derived NOEs, J-coupling, to guide your simulation? Why you made a different choice?
Yeah, it's a great question. So historically, the first reason is my collaborator is using SACS. And the second reason, I find SACS as an ideal system because it reports on the ion distributions, long-range and short-range interactions. And in an environment which you can tune the salt conditions and the temperature and everything you want. So it provides us a systematic studies on systems. I see that I don't see that level of flexibility in NMR, but NMR can definitely be a complementary approach, and we start basically also benefiting from it.
Yeah. If I can have just a quick second question. And the other thing I'm always wondering is with ions, cations in very highly charged systems. I mean, did you look at the binding times and things like that? What is your experience with the behavior of those cations with these very highly charged systems?
Yeah, so the cations are basically essential part of the folding. And some of the, so monovalent, there are two types of cations. One directly binds to the surface of the RNA in different regions, like the major groove and phosphate backbone. Some are diffusively present there, territorially there. They're not, they can exchange, but their place is always occupied by another ion. So the second group is almost impossible to measure. The first group can be measured by NMR relaxations, we can see them in the X-ray crystallographic data, cryo-EM data. Those exchange times for the specific ions are almost, for magnesium it's inaccessible in MD time scale, especially if it goes beyond one, if it loses more than one water molecule, the time scales are milliseconds that we cannot access to. But for diffusive ions, for magnesium, we found it to be accessible in the time scale of the MD simulation. So these are the magnesium ions that has a high radiation shell. They can come close to different cavities on the RNA surface and then diffuse back. So this is something we can accurately predict, and then we can compare ourselves with the X-ray data.
Thank you.
Thank you again.
Thank you.
So we got started a few minutes late this morning, so let's promptly be back at 11:10 so we don't fall farther behind schedule.